and welcome to this session on evolution. So we'll be seeing different aspects on this overarching theme in biology. And we're running a bit late, but in any case, we'll just move a bit into the lunch and poster sessions just to listen to these talks. So our first speaker is Abderraham Khila from the Institute for Functional Genomics in Lyon, and he will be telling us about development, selection, and species diversification.
So I feel like this talk is worth 3 talks because it's in front of 3 societies. So I'm very happy and very grateful to Miguel and to the organizers for this opportunity. It's my first time here in Gijón and my first time in this society. So thank you very much for the opportunity. So we are interested in phenotypic evolution in general, and we try to address this question by studying the interplay between mechanistic processes during development and processes that are governing evolutionary change. And we use this group of insects, we call them water-walking insects, as a model system. Before I go in more detail, I would like just to give a very brief reminder for some very basic concepts. So please forgive me if you are familiar with this. I will be talking about phenotypes that affect the concept of fitness. Fitness here is defined as the capacity or the average contribution of phenotypes or individuals or alleles to the next generation. And these are affected by 3 main components: survivorship, fecundity, and mating success. These components usually are affected by phenotype in asymmetric ways. For example, certain phenotypes that can be very bad for survivorship can be very good for mating success. And I invite you to go check Isabel Almudi's poster. There is a very striking phenotype that is specific to males that illustrates this example. So how do we tackle this question of origin of diversity? So this is a very old question, and it can be studied in many different ways. One way is mechanistic, and this is considering the phenotype as a central study object, and you can ask questions about what is the relationship between the genotype and the phenotype. In other words, what are the mechanisms of development that will generate certain phenotypes, and how can we associate changes through time of these mechanisms with changes in the phenotype? This is more of a how question. On the other hand, we can ask, what is the effect of the environment on this phenotype? So as you may know, development can generate an unlimited possibility of phenotypes, but selection sometimes favors some phenotypes over others. And we ask the question, why? Why certain phenotypes are better suited in certain environments than others? The group of insects we use to study these questions is a group called the Geromorpha. These are insects that live on the water surface. They are quite diverse, and you will find them everywhere except in the poles. So this group of insects, you can find them, they live in any type of water surface. You can find them in small reservoirs of water in the bromeliads. You can find them also hundreds of kilometers away from the shore in the open oceans. And you can find them everywhere in between. They are extremely diverse, and they have adapted to these different habitats, and they have diversified in a kind of a striking way. Before we started this work, what was existing is that there was a large body of knowledge and literature about the ecology, the biology, and the geography of these insects. So we had a good understanding of sexual selection, sexual dimorphism, phylogenies, fossil records, and these insects even attracted applied mathematicians and physicists that want to understand how insects or animal can walk on a fluid surface. What was missing is to basically turn these into an experimental model. So we brought them to the lab, we established cultures, and we started to import or optimize certain tools for the environmental genetic studies. The other reason why we are very much interested in this group of insects is there is a large variety of very interesting and striking phenotypes that one can actually ask these questions in. For example, they have this very particular capability of locomotion on a fluid substrate. And that is associated with a number of challenges that I will describe a little bit later. There is a good influence from interaction with predators that are different from the predators that their terrestrial ancestors used to encounter. There is a large literature and also a large number of phenotypes that are associated with sexual selection and sexual dimorphism. This, for example, this species here shows this striking phenotypic plasticity. These are two adult males of the same species, and you see one male is very, very large and another male is very small, and these are encoded by the same genotype. And you can ask, how can the same genotype generate such a diversity? And finally, there are phenotypes, for example, like this, very striking colored patterns in the eggs that we think have a mimicry function, or they scare away predators so they can avoid predation. So there is a wealth of phenotypes that one can actually use as a system to ask the questions I just laid down. So what is the advantage to make this transition from life on the water, from terrestrial life to life on the water surface? So one of the hypotheses is that there is unexploited opportunities in this environment because it's not accessible to all other insects. For example, here what you can see is the nymphs of a species called Geris, and they are feeding on a large cricket that you barely can see under them. So this means that prey that can be very, very large, when they fall on the water, they are defenseless, they are trapped, and that's what they eat, and that gives them an advantage. But this lifestyle is also associated with certain number of challenges. For example, how do an animal generate locomotion or movement on a substrate that is fluid? For us, when we push on the ground, there is friction, we can push and it's stable and we can move our body forward. These animals have to push on a moving substrate. And you can see in this video, there are waves that are going away from the body of the animal, so the substrate is moving back. One of the hypotheses we made when we started to notice this is that to live on this substrate, the animal must be fast, meaning that locomotion on fluid will select for faster speed. Here's another aspect. For example, these animals are always attacked from the bottom because they are exposed. There is nowhere to hide on the water surface. And the only way for them to escape predation is to be fast. So we wanted to test this hypothesis. And how do you make movement or locomotion faster? There are a number of ways. So these are the main parameters that will affect speed. You can be fast by increasing the size of the legs. You can be fast by increasing the amplitude of movement of the legs, or you can be fast by increasing the frequency of movement of the legs. So these are the main parameters that will determine speed, and these parameters are known to be in trade-off. For example, an animal that has longer legs will not be able to move them in a fast movement. So either one or the other. So I won't show you any details in this project. I'm just going to tell you the final result. Details can be found in this paper here. These animals managed to increase their speed by increasing all the parameters that affect speed. In other words, they broke all the trade-offs that are there on speed. So how did they do this? So if you look, so the work I'm going to show you in the next slide is mostly done by these three talented people. If you look at this species here, this is a very simplified phylogeny. Here you have an outgroup, a terrestrial outgroup. These are all water-walking insects, and you can see that there are differences in morphology. This species look more like this terrestrial outgroup, so it maintains or it retains an ancestral character state. Whereas if you move to these two derived species here, they have a very different body plan, where most importantly, the second leg is now the longest leg. And we will see in a second why that is. So if you look at the way of moving on the water, so this green species is this one here. So it basically moves on the water by walking, which is by alternating the movements of the leg, just like we do. Whereas this one here moves by rowing. These are two fundamentally different modes of locomotions that were enabled by the differences in body plan that has evolved in these different lineages. So this species here will generate movement only using the long middle legs, whereas this one here will use all the legs to move. So these species will be mostly in the margins of water bodies. It can go to the land or it can go to the water, while this one here is specialized in the water. It can no longer move on the ground. So how did these differences in terms of developmental mechanisms evolve in these species? So I'm going to show you how we move from this ground plan to this one here. So to cut a very long story short, Hox genes are what actually defines the morphology of the segments along the body. The difference between this animal, which is exclusively terrestrial, and this one, which can move on the water, is that this one here has legs that are 40 to 60% longer when you compare it to the body. So this morphology is made by a gene, a Hox gene called ulthrabithorax. This animal expresses UBX only in the third thoracic segment, whereas these two animals here will express UBX in the third thoracic segment and also in the second thoracic segment. So here we have a move or a deployment of a protein from one segment to cover another anterior segment. And this is a quite major genetic change. So this change explains the difference between these two morphologies, but it doesn't explain the difference between these two morphologies because they have the same pattern of expression and even they have the same dose. Third leg expresses much more protein than the second. When we look at the timing of expression of the gene, now we can see the difference between these two species. This one here, first UBX appears in the third leg, and it's only later when it appears in the second leg. Whereas in this species here, it's exactly the opposite. First it appears in the second leg, and later appears in the third. So it seems that differences in the timing can explain some of these differences in morphology between these two species. But this wasn't actually the only story. The function of the gene now also changed. So if you compare between these three species, in these two here, when we knock down the gene, the legs become shorter, all of them. This one, third leg becomes shorter, and this one, both legs become shorter. What was really surprising to us is that in this species here, which has the same pattern of expression, the knockdown will make the second leg shorter, but it will make the third leg longer. So we have opposite effects of the knockdown in the two adjacent segments. What we concluded from this study here is that these changes in morphology across these species have been encoded with changes in the timing, in the spatial expression, and also in the effect of the protein on the tissues of the leg. For example, the ancestral state, only one leg expresses UBX, and it makes it longer. In these two species here, both legs express UBX, the third leg expresses much more, and when we knock it down, both legs become shorter. So the function of the gene did not change at this node of the phylogeny. When we come to this lineage here, between these two, there is no change in the dose or in the spatial expression, except that there is a change in the way the leg tissue will sense the protein. At high level, the protein will reduce the leg, and at low level, it will make it bigger. So the way we've done it can be found in detail in this paper. So now, how does a protein, according to its level, affect two adjacent tissues in exactly opposite ways? So we looked a little bit in detail, and we did this analysis where we compare the transcriptome or activity or gene activity in the two legs, second and the third. What we found, and there's this gene here called GILT that is highly expressed in the second leg and is very low or not expressed at all in the third leg. When we look in the embryo, it's indeed expressed only in the tip of the second leg, and there is no expression in the first or in the third. So GILT and UBX have this profile of expression. When UBX is low, GILT is high. When UBX is high, GILT is low. So this suggests a repressive interaction between these two genes. And this is indeed what we found when we treat embryos with UBX RNAi. When we reduce UBX, then we have an increase in GILT expression in the second leg and also an ectopic expression in the third leg. What this means is that these differences in the dose of UBX between the two legs, it's actually there to modulate how much of certain target genes will be expressed. Some of them will be suppressed, but only partially in the second leg and completely shut down in the third leg. And when we modulate these levels of UBX expression, we can modulate how much of these targets can be expressed. And this is one way how UBX, through its dose, can actually regulate how much the leg will grow. So what does GILT itself do to the legs? So this is an embryo. GILT is expressed in the tip, which corresponds to the tarsus, which is what you see here, highlighted. It's a very long segment. When we knock down GILT using RNAi, we have embryos that look completely normal, except that they have very short second leg tarsi. So this is one way how UBX controls the size of the leg by controlling the targets which are in themselves important for growth. Or we can ask one question, so why is it important for the leg to have this morphology or this shape? So one of the, sorry, before I go there, we looked toward the phylogeny to see how this interaction between UBX and GILT, where it evolved. And what you see in this phylogeny here, there are 3 species that have the ancestral state, and these are 3 species that have the second leg very long. And it's only in this lineage here where we find this expression of GILT in the tarsus of the second leg, which means that this interaction between UBX and GILT is new, and it only evolved in this lineage where the function of UBX became dose-dependent. So now the question is, why is it so important for the animal to have this particular morphology? And that's because not only the legs are important for locomotion, they are critical for avoiding predation. And to go back to my fitness diagram, this is directly related to the survivorship of the animal. So here, a fish is going to attack, and what the water stride is going to do is going to use its leg, it's going to jump really high, and then it will fall back and escape. This is a high-speed camera video. In real life, we don't see anything. We only see the water being disturbed. So we wanted to test this. So what we did actually is we took animals, we treated them with GILT RNAi, and we generated animals that have approximately 20% shorter second leg. And we tested how well they are at making this behavior. And this is what we got. So this is a control animal. This is a GILT RNAi animal, which has approximately 20% shorter legs. And you can see that the control animals are much more, or deliver much higher performance, or much more efficient performance in jumping, which is a behavior that is very important for escaping predators. So again, controls, etc., can be found in this paper. So one take-home message from this part is that evolutionary change can happen by just using and reusing pre-existing developmental programs. Here we see the case of one very pleiotropic, very important Hox gene, which is an upstream regulator, and its pattern of expression, its dose, its timing of expression, everything changes throughout the phylogeny. And those changes are associated with the changes in morphology, which in turn are associated with the lifestyle, the kind of selection each of these animals encounters. I want to tell you another story that this time involves a different type of phenotype. This is a phenotype that we call an evolutionary innovation or a novelty. These are usually phenotypes that evolved de novo in certain lineages and that we cannot trace by descent. In other words, that the ancestors of those lineages don't possess these phenotypes. And this is a very important question, and it tells us a great deal about how the evolutionary process works. So one of the questions is, where do these phenotypes come from? What are the gene networks that build them? What is their importance? What did it do in nature for these lineages, etc.? So the phenotype, so this project was conducted by a former postdoc. She's now in Cambridge, Emilia Santos. And the phenotype I'm talking about is a very specific structure that you find in the tip of the legs of one of the water striders. So if you look at these two species, Ragovelia and Stridovelia, they are very closely related, and they both use this derived mode of locomotion through rowing. The difference between these two species is that if you look in the tip of the legs of Ragovelia, you have this structure called fan, whereas in Stridovelia, you don't have such a structure. This structure exists only in this genus, and it doesn't exist anywhere outside the Ragovelia genus. And all the Ragovelia species that we know, approximately 200 species, they all have it. This is how the fan looks like. So now when you dissect it and take it out of the leg, these are the claws, these big structures here, and the fan is this very complex, very nicely neat system that is formed of these plumes. They have primary branches and these very thin secondary branches, and it's actually a structure that the animal can deploy or retract. If you look closer at this, at the tip of the leg is forked and there's space for it to be parked or deployed. Well, the question we asked is, first, how do you build such a new structure during development in these species? So most of the animals we work with, we don't have genomes. So we always resort to comparative transcriptomics. So what we've done, we took the three legs, first leg, second leg, and third leg. The second leg is the only leg that has the fan, and we sequenced the genes that are active in these legs, and we were looking for a group of genes that would be highly expressed in the second leg, but not in the third leg or in the first leg. We obtained approximately 90 genes that are expressed highly in the second leg, but not in the other legs. And we classified these 90 genes in terms of expression pattern based on in situ hybridization. So basically, when we've done our transcriptome, it was the entire leg. The fan only develops at the tip of the leg. So those 90 genes don't, some of them probably will be expressed in different cell populations than the ones that make the fan. So we wanted to know if we have genes that are expressed exactly where the fan appears. And so this is what the pattern would look like, or this is the pattern we were looking for. No expression in the first leg and in the third leg, and a very localized expression at the tip of the second leg. And that is very striking because it is directly associated with the part of the leg where the fan will appear. So such gene, we found 5 of them. And here I'm only showing the ones that are expressed only in this cell population and nowhere else in the leg. So what was striking to us is that among the 5 genes that show this pattern, 3 of them are quite well known. One of them is the gene yellow, which is known to be important for cuticle formation and for pigmentation in the cuticle. So that was not surprising for us. 2 of them was cuticular protein genes. That also is not surprising because the fan is primarily cuticular structure. But 2 of those genes were genes that were not known before. When we looked closer, they were actually paralogs. They are very similar in sequence, and when we rebuilt the phylogeny using these sequences, what we found is that this sample of species that are all also geromorpha have only 1 copy of these 2 paralogs. And these are 3 species of the Ragovelia genus. They all have fans, and they all have 2 copies of these 2 paralogs. And what this allows us to do is to map an event of gene duplication that happened exactly at the time when the fan evolved in this lineage. So now we have 2 very striking events. One of them is a gain of regulation or expression of these 2 paralogs in the tip of the leg where the fan will form. And the other one is a mapping of this gene duplication exactly where the fan appeared during evolution. So we wanted to know what these two genes actually do. So before I go any further, we decided to call these two genes geisha and mother of geisha, and that's because of the analogy between the fan and these geisha fans that you see with these Japanese entertainers. So geisha being the new copy, mother of geisha is a copy that is more similar to the copy that other species have. When we knock them down using RNAi, this is what we get. This would be a control fan, and this is an RNAi, a fan from an individual treated with RNAi. You see that the claws are completely wild type, just like the control, whereas the fan is deeply reduced. And this is actually very striking because it means that these two genes control the development of the fan and nothing else in the leg. What was even more striking is that when we actually analyze the way the animals use their fans after we do RNAi, they do it exactly the same as the controls. So this is a control animal. If you follow the arrows, you see when the fan is going to be deployed. So here, you see it deployed. And the RNAi animals will do exactly the same thing, except that they don't have anything else. All you see is the little claws coming out. This is also very important because it means that the RNAi experiment affected the fan, but it didn't affect the muscles that move the fan, and it didn't affect also the nerves that are important for moving the fan. So why do these animals need a fan in the first place? If you go to the field, this is what Ragovillia does, or this is its lifestyle. It's a species that lives in fast-flowing streams, and they have this lifestyle that is permanent movement. They don't stop. They do everything while they're moving this very fast. Whereas this closely related species, Stridulophilia, you'll find it a lot of times in the same streams, except that it will be on the side, always associated with substrates and plants, etc. So they are very fast, but they are most of the time static, and they will only move from time to time. So we thought that this lifestyle is probably what has favored the evolution of the fan. So to test exactly what the fan does for this species, we devised an experiment where we have 4 groups of individuals: Stridulophilia, which is the control out group that doesn't have fan; control Ragovillia with completely intact fan; groups of Ragovillia where we surgically remove the fan; and then the group of RNAi individuals where there is not a complete fan, but there is some left, or some pieces of the fan left. We took these individuals and we tested them in a calm water setup. So this is calm water where this species would normally live. And what was really surprising to us is that the species without the fan is the fastest in calm water. And these 3 groups, no matter what the condition of the fan, they are all as fast. That means that the fan is not necessarily important to make them faster. Although what was important also is, if you look at stroke frequency, this one is faster because it strokes the most. So it works very hard to generate that speed. And for the fan individuals, the one with intact fan strokes the least, and the one where we do RNAi is intermediate between this one and the one where we surgically remove the fan. So it seems like the fan is a device that saves energy rather than makes them be fast. Now what we did is we devised another setup, but this time in stream environment. This is where this species will actually specialize. So you have a canal where there's a current that we can control speed, there's a loser bucket and a winner bucket, and we can actually count how many individuals make it here and how much time it takes them to make it there. This is how the setup looks in real life, and then the individuals will try to go against the current to the winner bucket. When we analyze this group of individuals in this setup, now this is a very striking result. This species, which is the fastest in calm water, 0% of them were able to go against the current. So without fan, they can't do it. The ones which have intact fan, they all make it against the current, and they make it in a very short period of time. And again, the ones with pieces of fan, they have an intermediate performance. 78% of them made it against the current, and it took them less time than the ones where we removed the fan surgically. So what this means is that this evolutionary innovation has evolved because probably it has been favored by this very challenging environment, which is this fast stream water. And this is one way of how species can actually expand their habitat and expand their specialization and diversify in nature. So this is just my last slide. Another also take-home message is not only phenotypic evolution happens through reusing the same developmental programs, but sometimes also new developmental programs can emerge to make these kind of structures. So with this, I'd like to thank you again for your attention. I'd like to thank the people of the lab, especially the ones I mentioned earlier that are involved in this work, and I'm happy if there are any questions.
Hi, wonderful talk, amazing. So I was wondering, in your gene application of geisha, do you know what are the regulatory changes that are causing the misexpression of the gene or the specific pattern?
No, we don't know yet. We don't know yet. We know that there is new regulation because when we look at the other species that don't have fans, none of them expresses the one copy they have in the legs. Actually, it's even striking. We take probes from a different species that doesn't express. When we use it in Ragovelia, we have the pattern. So we know that they don't express it, therefore there is a regulatory change, but we don't know what it is.
Fascinating talk. So about the mechanism why the length, the leg gets elongated, do you know whether it's more proliferation, more initial cells? What is the mechanism of the elongation?
We do. I should have put a couple of slides to the cell biology colleagues. It's more leg length is more cell proliferation. Size of cells doesn't change. When a leg is longer, it's because it has more cells, and we quantified that. And actually, UBX controls this process.
Is there any case where you get extra segments instead of a segment that is longer?
Never, no, never.
Have you looked at all these changes in the leg for those changes in the UBX locus?
Have I looked at the changes in the legs in association with sequence change in the UBX locus? We have in the coding sequence.
And non-regulatory regions?
No, our first genome became exploitable just a couple months ago, and we have a second one just came. So we should be able to start looking. In terms of coding sequence, all the domains that you'd expect to find there are there.
Okay. Any other questions? Just last one over there.
I have a question regarding, I'm here, regarding the difference you make between the evolutionary innovation you're saying and the tinkering of existing functions. But in the second example, you have a duplicated paralog that does the new function, but you don't know what the ancestral mother is doing in the other species. So maybe you're actually in the same situation where you have a function that has been sort of modified specifically in the lineage, but that is reusing something functional.
It is possible. We tested the function of mother of geisha in the other lineages, and it doesn't seem to have a developmental function. We actually, in terms of RNAi and also in terms of expression, there is no developmental function. Although we find it in the transcriptomes, that means it's expressed somewhere. We don't know what function it does. It's clearly not developmental. because that's the easiest to detect. Thank you.
Okay, our next speaker is Patrick Fitze from the Museo Nacional de Ciencias Naturales, Madrid. And he will talk about sexual selection. In his speech is entitled Drivers of Sexual Selection and Consequences for the Maintenance of Genetics Polymorphisms.
Perfect. Good morning. First of all, I would like to thank the organizers for inviting me into this very nice conference. When we think about evolution, we generally think about three mechanisms that lead to evolution. Namely, genetic drift, natural selection, or sexual selection. Darwin was the first to precisely describe the concept of sexual selection in his famous book on the descent of man and selection in relation to sex. His definition was that sexual selection is the relationship between a trait and its effect on fitness through sexual competition. And surprisingly, nowadays, the definition is still the same. However, also Darwin described sexual selection a long time ago. Its importance for evolution is still under debate. Here you have some examples of these birds of paradise, where male display these very nice, fancy plumages, while the females are drab. And it's suggested that this evolved due to sexual selection. So the question is, what is sexual selection? For that, I start with this very simple example, where we have a population, or we assume a population where we have only 3 females. These females are exactly identical. And then we assume that there are 6 males, yet only differ in size. We assume that's a polyandrous species where females mate with 2 males. And now, if we think about random mating, what we can then predict is that, on average, each female mates with 2 males and each male with 1 male. And that means we will have this following outcome, where we have the number of offspring per male, what links to, say, fitness, what the previous speaker was explaining very nicely in his introduction. And then you see there is no difference between the males. If there is non-random mating, then we can have such patterns like you see here, where 1 male suddenly mates with all females, and the outcome is a skewed distribution. If this skewed distribution is due to sexual competition, then we talk about sexual selection. So how can patterns arise due to competition? Well, the first possibility is that females prefer big males. We would call that female mate choice. Another alternative hypothesis is that males fight for access to females, so we have male-male competition, and we have this pattern. Of course, this case is too simple. The general pattern is that, of course, females differ as well. And then we have 2 additional possibilities, namely that females compete among each other for access to males, what we call female-female competition, or that males evolve preferences, what we call male mate choice. So what are the consequences of sexual selection? Well, the consequences are conflicts. These conflicts are among males, among females, or they are between the 2 sexes. If the conflicts are between the sexes, then we talk about sexual conflicts. And these conflicts can be either symmetric or asymmetric, and that means the costs are either paid by both sexes or by only one sex. If we look at a classic example of sexual conflict, for example, the conflict about the number of eggs per clutch. We see that there is an optima here in females, and in males the optima differs, and this is because females usually produce more gametes. Yes, the female gametes are bigger, females are limited in egg production to the size of the body cavity, etc., etc., and thus their optima is smaller. What happens if we have two different optimas? Well, two different optimas means there is scope for the evolution of strategies of one sex to move the optima of the other sex, or to move the reproductive investment towards its own optima. The male strategy is to try to make sure that the females produces bigger clutch sizes, while for the females, of course, the optimal strategy is to be in the optima. Besides this general conflict, there's as well an important other conflict, sexual conflict over the number of mate partners. And there you see the pattern is different. Oh, sorry. So if we have this, I have to just switch. So we call, if we have this different optimas, where both sexes steer in the wrong direction or the other direction, then we talk about the tug of war between females and males over optimal reproductive investment. Another sexual conflict is the conflict about the female's number of mate partners. For a male, the best thing is if the female only copulates with one male, with one male, because then he fertilizes all the eggs. However, for a female, this is not the case, because females are limited in the number of eggs they can produce. They only have two possibilities. The first possibility is to choose a better mate, and the second possibility is to mate with more mates. And here, of course, there is another example of sexual conflict. Sexual conflict can affect trait evolution, and this has been nicely shown in Drosophila melanogaster. So males transfer sperm and seminal fluids during copulation, and Chapman and al. Showed that males actually transmit toxic seminal fluids that reduce a female's lifespan. So if we have the two optima, what happens is that with more males a female mates, the less she survives, and thus the optimal strategy is, in this case, to mate with less males and produce bigger clutches. What is exactly what is beneficial for males. If we talk about the tug of war, then the tug of war is won by males. But this is of course only temporal, because selection will favor females that evolve resistance against these toxic seminal fluids, and that's exactly what these authors observed. And that means that the optimal number of, so the optima moves towards, or the investment moves towards the female's optima. Once the investment goes towards the female's optima, again, there is scope for the evolution that males evolve something new that starts to move the average towards their own benefit. And what they found is that males evolve more toxic seminal fluids. That means females, again, win the tug of war, and we can continue that over generations. And the big question, of course, is what parameters affect the strength of sexual selection? What parameter affects this sexual conflict? To this end, we run experiments in the European common lizard that inhabits as well the mountains just here in the north, in the south of Gijón. And we established populations of this lizard where we can manipulate principally everything. So what we did is we produced populations that were female biased and populations that were male biased, and they did not differ in any other traits. We followed up these populations over 1 year. After 1 year, we look at the reproduction. We genotype all eggs and offspring, and then determine the number of fathers that fertilize the eggs of a given female. Here you see the results. So here you have the female's body size, and here the number of males that fertilized her eggs. And in female biased populations, what is this blue line, you see there is no selection going on the level of female body size. Eggs were followed by approximately 2.5 males. However, in male-biased populations, the number of fathers increased with female body size, reaching up to 4 fathers in the biggest females. That means population sex ratio affects a female's number of mate partners. When we look at reproductive success in terms of number of offspring per clutch, then you see that in the female-biased populations, the mean was much higher, namely females produced on average 2 more offspring, what is surprising given that the maximum is 10 offspring. Up here you see a mating struggle. So a mating struggle, the males usually bite the females on the posterior abdomen and then try to copulate, what looks like a struggle, and what produces injuries on the female. When we quantified the injuries, we found that females in male-biased populations had up to 3 times more injuries than females of female-biased populations. And when we look at survival, first we look at survival in males. You see that there are no differences between female and male-biased populations, neither in juveniles, nor in yearlings, nor in adults. If we look at females, in female-biased populations, you see that these means are almost the same. However, in the females of male-biased populations, the survival was very low, below 20%. There was a huge effect. So what does that show us? Well, this shows that males increase a female's mating rate at the expense of females' clutch size and survival. And that means it's a classic example of sexual conflict that is determined by the population sex ratio. When we think about the speed of evolution that could be imposed, for example, as sexual selection, then everybody would rapidly say, well, that's not what we should analyze. We should look at, to understand the speed, we need to analyze the intensity of sexual selection. And there are 2 competing hypotheses. First, there exists the population sex ratio that determines the intensity of sexual selection and the costs of reproduction. In the previous example, we have seen that the costs for a female in male-biased populations is very high. They reduce the number of, have a reduced number of offspring, they have more injuries, and they they survive less. So from that we can derive predictions for both hypotheses. First, for the operational sex ratio, we would predict that the intensity on males is bigger in male-biased populations. Why is this the case? Well, there's many males, few females. Females can choose among males, and that means there is more sexual selection going on males. From the cost of reproduction point of view, we would predict that in male-biased populations, the intensity of sexual selection on males is lower than female-biased population because females in male-biased populations bear a lot of important costs. So due to this cost, they can no longer be selective. When we look at predictions for females, we predicted that under the operational sex ratio theory, sexual selection on females would be higher in female-biased populations than in male-biased populations because there is many females, not so many males, and most likely not all males will be able to, or not all females will be able to copulate. For the cost of reproduction, we just predict the contrary. If we look at the results on sexual selection on males, you see different slopes. The steeper the slope, the more sexual selection or the more intense sexual selection. As you can easily see, the blue slopes were steeper. That means intensity of sexual selection on males was stronger in female-biased populations than male-biased populations. And when we look at the sexual selection on females, you see that in female-biased populations, this relationship was flat, while it was a positive slope or even a very positive slope in male-biased populations, what means that sexual selection on females is higher and stronger than in male-biased populations than female-biased populations. To conclude, these results here coincide with our cost of reproduction hypothesis. And that means this experiment showed that the relative cost of reproduction indeed determined the intensity of sexual selection and thus the speed of evolution. So, next question. Does sexual selection necessarily lead to evolution? If we look at the theoretic models on sexual selection, almost all of these models show that sexual selection leads to the evolution, more faster or less fast. Typical example is Fitze's runaway process. There are only very few models that suggest that not necessarily that it's not necessary that there exists evolution due to sexual selection. Namely, the sexual selection mediated by rock, paper, scissors models. Solid evidence is lacking, and if we talk about rock, paper, scissors games, we are talking actually indeed about this game that is played by kids, where you have rock, paper, and scissors strategies, and where none of the three strategies wins in all contexts. So theoretical models show that if the strategy is genetically linked with the reproductive success, the evolution of rock, paper, scissors dynamics is possible. Here you have such a model output, where you have the generations and the frequencies of the three strategies. And the model predicts that if scissors become frequent, they lose the advantage, and then rock gets a benefit and starts to get frequent. Once rock starts to get frequent, it loses its advantage, and paper has an advantage and starts to get frequent. And once paper starts to get frequent, it loses its advantage, and again scissors starts to have an advantage and increase in frequency. Such rock, paper, scissors dynamics have been described first in the side-blotched lizard. Here you see such a frequency cycle. Each arrow is a frequency change from one year to the other. In these three male colomorphs, orange, yellow, and blue. And such cycles have been found later on in quite a few species, namely in organisms, say, in bacteria, in insects, in plants. What is common to most of these examples is that they're short-lived, that there exists no generational overlap. There's only one exception, and this is the European common lizard, where males exhibit three morphs, orange, yellow, and white, where rock, paper, scissors-like dynamics have been shown. And for simplicity, I will use always the same topology of this ternary plot, and the direction that has been observed in nature is contraclockwise. That's what you need to keep in mind for the rest of the talk. So males survive between 4 and 6 years. Survival is independent of colomorph, and the immature males that are between 1 and 2 years old are not yet reproducing. They're in competition with adult males, and they are suggested to produce the changes in frequency cycle. As I always said, the populations, also the generations, are largely overlapping. The theoretic models actually showed that simple mechanisms that have been assumed to drive these games cannot generate enough frequency-dependent selection to explain the rapid cycles. And what they suggest is that very complex mechanisms evolved, namely frequency-dependent selections generated by sexual selection and additive subsequent survival selection. Well, it's quite amazing. And most scientists would have said when they saw the models that this is simply not possible. So we went out to test whether these models indeed, whether there's indeed some basis for these models. To this end, we produced, again in our population system, populations that had absolutely identical adult male frequency between white and orange, and then we produced two treatments. We simulated the RPS phase advanced towards orange, and RPS phase regressed towards white by releasing either immature males with orange colomorph bias or with white colomorph bias. What did we find? Well, we followed them up for a year, we genotyped the offspring, and when we looked at the success of the different male morphs, and we found that in phase advanced towards orange, yellow-white males had the highest siring success, and that means that the phase advances towards yellow, exactly what we would predict because it's counterclockwise. In phase regress towards white, what you do not see in nature, but we simulated to see what happens, we found that orange had the highest success, and that means the phase advance goes towards orange, counterclockwise again. And these results show, first, that adult male polymorph is indeed sexually selected, second, that sexual selection is congruent with the frequency cycles. The second prediction was that there needs to be a second mechanism producing enough frequency-dependent selection. So we looked at survival selection and found that offspring of yellow-white males and offspring of orange-orange males had the highest siring success. That means that the survival of the offspring of the morph with the greatest siring success was significantly greater than of the offspring of the other morphs. And that's perfectly consistent with the existence of two additive and temporally separated types of frequency-dependent selection. So it's the first proof for the evolution of this highly complex evolutionary rock, paper, scissors dynamics. And what it means is first that sexual selection can maintain genetic variation over time, and second, this clearly contrasts to the classic models of sexual selection that suggest that there needs to be always evolution. Given that there's major generation overlap in these species, it also suggests that rock, paper, scissors dynamics can maintain, or could maintain genetic variation in quite a lot of species, namely in all species with sexual selection and even in species with overlapping generations. So if we look again at the model predictions, you see that these frequency cycles are very regular. Well, rather regular if you look at the ternary plot and we plot these predictions, and we will predict such a circle here. But actually what we found, or what was described, is not at all regular circles. The big question is, why are they not regular? One hypothesis is that climatic condition might be responsible for that. And we carried out experiments to test this hypothesis. So we produced generation with yellow, white, and orange bias, and we submitted them either to less or to more precipitation, and then quantified their effect on survival selection. In juveniles, you can easily see there's no difference at all. However, in immatures and adults, individuals of white bias populations exposed to more humidity had reduced survival. So that means that survival indeed depends on the color morph frequency and the environmental conditions. Interestingly, when we looked at the color morphs, whether they survive differently, we found no decipherable differences among male color morphs nor among sexes. And that means that the color morph frequency or the genetic characteristics of a population actually affect the fate by means of natural selection. And this is congruent with environmental conditions that affect the orbits of the frequency cycles. Here we used only one parameter. You can imagine there's plenty of parameters in nature that can feed back. However, there were more results. In yearlings, we found that females grew faster in population with more precipitation. What does that mean? Well, that means that females became earlier mature. If we look at effects on reproduction, females produce different sex ratios, and these sex ratio differences, there were sex ratio differences among color morph treatments. And that means both of these results change the population sex ratio. So this means that color morph frequency and environmental conditions both affect sexual conflict. Or with other words, that natural selection feeds back into sexual selection. So to wrap up, the parameters that affect the intensity of sexual selection are the costs of reproduction, sex and age structure of a population, color morph frequency, or more generally, the genetic characteristics of a population, and environmental condition. And with that, we try to respond to the question, not what is the speed of evolution, but what determines the speed of evolution. With that, I would like to acknowledge my co-authors and agencies that founded the project, namely the Ministry, the Spanish and the Swiss Ministry of Science. And last but not least, you for your attention. Thank you very much.
Thank you, Patrick. Is there any question?
So in the experiment where you have male-biased treatment, you have large females that are, their mating frequency increases. Does that mean that males tend to choose bigger females? And what is the size of the males that mated with those females?
So
they're also large.
Yeah, so there are several questions. First, there is subtractive mating going on. So generally, if population sex ratio is more or less balanced, you have a quite good link between the size of the males and the size of the females. So in this species, the males are smaller than females, because males will copulate with the biggest females. But what happened here is that the biggest females were the most attractive ones, and we have evidence that males indeed choose, if they can choose, the big females. Why is this the case? Well, they have more eggs to fertilize. With the same effort, more eggs fertilizable. And if the females are not under pressure, so if there are no, say, sexual aggression imposed, then a small male has no chance to copulate. However, if there's a lot of sexual aggression going on, so if they impose enormous costs on females, then females suddenly are less selective, because they cannot afford to resist.
Hi, Patrick. One question out of ignorance. To which extent does the probability of encountering a male with a female will affect the conclusions that you draw from these experiments on populations that I guess the probability of encountering is very high? I'm thinking about species that live virtually in isolated individuals, and a male sees a female 3 times in his life, perhaps.
Yes, it's a very interesting question, and it goes to the heart of all this series, because first, this is a species with extremely high populations. So you can imagine in a space like this tribune, you would have, I don't know, 200-300 individuals. So it's very easy for them to encounter each other. But there are, of course, populations that are extremely small. And there are only a few, would be only 3 individuals. So there you have these problems. We would talk about the so-called Ali effect. And one of the possible problems is to encounter. Of course, if a female cannot select, there are only 2 options. Either to make a minimal reproductive effort, that means they anyway produce eggs even if they have not seen males, that means everything's for nothing, or just copulate with this only possibility. And what we found and was shown is that the smaller the choice for a female, the less chooses you will be. And that's described in many species, including humans as well. And it goes in 2 directions, okay? If I say females that choose, we have to keep in mind that males choose as well. Almost in all species, probably. But it's exactly the same pattern. And you can see that's very similar with the congruence in the results. If you have more sexual selection on males, then automatically you need to have less sexual selection on females, and vice versa.
Thank you. Patrick, you told that environmental condition may change sex ratio, and I suppose this is especially important in species that have a temperature-dependent sex determination. And is there also any evidence that the climate change may be affecting sexual selection?
Good question. So first thing, I don't think that this is related only to poikiloterm species. So can be in all. So imagine a bird that flies around, but it's raining all day long, and if rain is bad for the bird, then he has not a lot of choice what he can do. He cannot fly 10 kilometers to copulate with given male. And so I think it's probably quite a general thing that climatic conditions feed back. This was actually one of the first demonstrations that this happens, and that of course has important implications. So if the temperatures or the climatic conditions change, this will change sexual selection. I did not present the results, so we did other experiments where we find actually that different climatic conditions change the strengths of sexual selection. That means the speed of evolution.
Thank you very much.
Thank you.
Our next speaker is David Posada from the Universidad de Vigo, and he will talk about genomic evolution of intratumoral.
Okay, hello. I would like to start thanking the organizer for the invitation. It's a real pleasure to be here. And I'm going to finish this session now talking about evolution also, but in a different context, which is at the cell level. Okay. Vale, muy bien. No, pero no está. Okay, here we go. So basically, most research in evolution has been in germline evolution. It's about descent, families, individuals, through generations. But indeed, there is a process of evolution that is happening inside each body, and that is called somatic evolution. You can think that in every mitosis there is some mistake. So on tumors, we believe that the average number of errors in a single mitosis is 1 to 3 mutations. So we will not have any identical cell in the body, okay, regarding the whole genome, at the whole genome level. So we do have different mutations and different genomes in different parts of the body and different cells. They might matter or not, okay? These mutations, most of them indeed are irrelevant for in the functional aspects, and in the cancer arena, these are called passenger mutations. But other mutations do change the function of the cell. And we know, all of us know very well, one of the most of the dramatic effects of this somatic evolution, functional changes, which is called cancer, which is one single cell gains new abilities to do several things like growing faster, attract angiogenesis, and move to distant places like metastasis. So basically, all starts with an accumulation of mutations from a healthy cell. At some point, 1, 2, or 3, a number of mutations. We believe that not that many. Okay, the latest hypothesis, like on average 5 mutations, might be like a reasonable number. It's still discussable, like a common number, if there is something common in cancer. Basically, you acquire, in this moment in time, a single cell becomes malignant, okay? Grows, and then we have a clone. Okay? A clone, in my definition, is a set of identical cells. They have the same genome. Okay? It doesn't mean that different clones have different functions. Basically, the colors here are different clones, different genetic configurations. So basically, what happens now is a process of evolution. The paradigm so far in cancer is that this is a process of Darwinian evolution. We have like a blue clone. Suddenly there is new mutations that might change to a slightly lighter clone, a green clone, and then we have selective pressures at some point. Okay? This might be physical pressures, they might be competition with other cells, whatever, and that only this clone is able to grow, outgrows the other clones. Okay? This is positive selection, keeps evolving, becomes bigger, at some point acquires, for example, the ability to colonize new ecosystems. That would be a metastasis, to change places, okay? And another some point, there might be another selection pressure like therapy, and only, for example, this little guy here, a minor clone in the population, is able to resist and produces a relapse. So this whole process from a healthy cell towards a whole ecosystem of different tumor cells, okay, is a process of evolution. And the paradigm that this is Darwinian evolution, dominated by positive selection. Okay, that's the main idea. Well, all this process unavoidably conducts to two levels of heterogeneity. First, different individuals have different genetic changes in their tumors, even for the same cancer type. Okay? A cancer type is basically the tissue of origin. In some cases, the cell of origin, but normally the tissue of origin. They share some mutations in some cases, but they don't in others. Okay? These mutations, which are called driver mutations, in driver genes, the genes responsible for cancer, P53, KRAS, all these oncogenes, tumor suppressor genes, they not always appear recurrently in all genomes. Okay? Most effort in cancer genomics has been to identify repetitions. So if you look at colorectal cancer, which is the gene that is commonly mutated in colorectal cancer. So might be APC, might be KRAS, P53, but no single gene is mutated in all cancers, from a single type even, okay? So there is variation between individuals. And of course, the process of selection, this competition, also provokes that within a tumor, there is intratumoral heterogeneity. We have different clones. Now we know this, the more power we have to explore the genome, okay, sequencing technologies, the more we see that there are many differences among different cells in the genomes of the tumor. And we also know that there are functional differences in these clones. Well, this is quite relevant if you think about it, because when you take a biopsy from a patient, you're going to pick up one of these colors, one of these mutations, and not all of them, okay? And therefore, you're going to get only a biased representation of conclusions about your tumor. This might be relevant in some cases and in others not, but we need to be aware of this intratumoral heterogeneity. So I am an evolutionary biologist that only recently, 3 years ago, started interested in understanding cancer. Okay, cancer, tumor, intratumoral heterogeneity, which are the processes behind this generation of heterogeneity within a single tumor. Okay? In our lab, we focus on two types of cancer as models. Okay? One is colorectal cancer, which for me is a terrestrial organism, and the other is a leukemia, chronic lymphocytic leukemia, which for me is a marine organism. Okay? And we do have different expectations about the evolution. How do we sample this intratumoral heterogeneity? The common approach is what is normally known as bulk sequencing. Basically, you get a biopsy, okay, a piece of tissue, which may be 1 or 2 centimeters, okay, cube, and basically there you will have around 10 to the 9, 10 to the 12 cells, okay, in that little samples. So we have billions of cells, yeah, in a small piece of tissue. And you just sequence the bulk of that, the whole thing. Okay, that's the typical, that's the more common approach, to get this tissue and you sequence the whole thing. But I just told you that of these billions of cells, there are differences between them, okay? So this type of approach gets a consensus, okay? The image we get from that type of approach is a consensus of that piece of tissue which is a part of a tumor with different colors, where colors are mutations, okay? Another approach which normally is more impractical in the clinic, okay, it's more difficult to get a pathologist to give you different samples from the same tumor, is multi-regional sample, where what we do basically, this is a bulk, so you take a single piece of tissue. In a multi-regional approach, you take different bulks. Well, this is better because you're going to be able to represent more accurately the landscape of genetic mutations in that tumor. And this is what normally evolutionary biologists do when we go to different archipelagos, okay? And we sample different populations of individuals in different beaches in this case, okay? And that's what we call phylogeography. So what I want to do is to understand the phylogeography of tumors. Treat tumors and cancers as archipelagos. Treat metastases as islands. Okay? treat the primary tumor as a continent from which individuals jump and move. So we have 100 years of experience and techniques for studying, understanding genetic variation in space, and I strongly believe that this can be applied to cancer. Okay? So that's what I'm trying to do. But this is not easy, because when we go to Cape Verde and sample conus, we can get individuals. Getting individuals in cancers means getting cells. The individual in a population is a cell in a tumor. That's the homologous level, okay, a single cell. Now we have single cell techniques, okay. So our current grant is ERC grant to use single cell approaches, whole genome single cells, because the mutation rate is low, and I want to apply statistical techniques from population genetics, phylogenetics, and phylogeography. I need many mutations, so I need to explore the whole genome. Unfortunately, this is not that easy, okay? And I was hoping to have here already some data for single cells, but we just did an experiment with 150 whole genomes from 150 single cells, and nothing worked, okay? Everything was working really well for cell lines, but once we move into the real cell, the patient cells, we're having trouble, so hopefully next time. So sorry, so I'm gonna focus today on this approach, okay? On the multi-regional approach. I do really think that single cell is the hope for understanding from an evolutionary point of view cancer. But we can also do things with multi-regional sequences, but there are limitations, as you will see. Well, this is work from Joao. Basically, I'm going to present several studies, okay? Because what we do, instead of the most cancer genomics, what they do is to get thousands of tumors and look for recurrence, okay? That's what I said before. You get thousands of tumors and you see which mutations are common across a cohort of 2,000 tumors. We do the opposite. We just want to study in detail few individuals. It's a question of money, basically, because sequencing a whole genome, we want to sequence hundreds of whole genomes from single patients, okay, from different organs. That's what we're doing. Basically, to get our feet wet, we started with this sample. This is an autopsy where we got 61 samples from a single individual from colorectal cancer. And we already sequenced, we started doing whole exome sequencing, okay, NGS from 20 locations, as you can see described, right? So we got locations from the primary tumor, okay, those in red, healthy samples. When we study cancer, we always care about somatic mutations, those mutations which are in the cancer cells, but not in the healthy tissue. Those are the somatic mutations, mutations which are de novo. So we remove all germline variation, okay? And for this, we need a control, and the control is healthy tissue. So this is always a paired sample, healthy and tumor for every patient. So we sequence this, in this case with ion torrent, okay? And, well, after doing a lot of quality control, removal, if you work with NGS data, you know that you have to be very careful about your colors and so on, but I'm gonna afford you all the details, all the gritty details. Basically, we need to get rid of copy number variations. In cancer, there is a lot of structural variations, right? So you get pieces of chromosomes duplicated and moving and broken. So you get, in an advanced cancer, a cell is a mess, a tumor cell is a mess. Okay, you have chromotripsis, you have many events. We just want to focus on single nucleotide variants, because there is when where we do have statistical models to understand the evolution of A to C, A to T, A to G, okay? And basically, we just get rid of copy number variations, like here, here, there, and there. And at the end, we have a golden set of almost 2,000 single nucleotide variants, okay? For this data set. Here, well, just as a sanity check, we just check that we find mutations in the expected genes, in the driver genes. These are the driver genes for colorectal cancer. APC is one of the first ones, KRAS, then comes P53. And we do find in most of them, but not in all. But most strangely, look at this pattern. We don't need to care about the labels. Basically, this is which mutations we find in each region. And here, so what you have, in the rows, you have the different geographical regions, and in the columns, you have different mutations. So you can see that this pattern is variable. There are some mutations, only a few, one really that appears in most locations, okay? The second one appears in 6 locations, but you know, these mutations only appears in 2 regions. But there is a lot of variation. So different regions have different driver mutations, okay? And that's very relevant, Because if my biopsy takes this sample, I'm going to believe that this tumor has these mutations. But if the biopsy is this, I'm going to believe that the tumor has other mutations. That's a big trouble, but it's the way it is. And this is not easy to solve. Okay? We need to be aware of that there is a lot of heterogeneity among the geographical location of driver mutations in tumors. The diversity, the levels of diversity, you might think that they might be different in different regions, more diverse, but actually they are very similar, okay? The heterozygosity across the different regions, which somehow suggests that this diversity might be old and there is a lot of mixing. So cells, at least in this case, are really well mixed in the tumor. We don't have a clear population structure, as we'll see next. We do find different, in cancer is very important, the context of a mutation. So the probability of changing from C to G depends a lot versus C to T. The relative probabilities of substitutions depend on the sites you have 5' and 3', you know, below and after you, okay? Before and after you, sorry. And we call that mutational signatures. Okay? So we do have special mutational signatures like this. These are the triplets, okay? So we have different proportions for different parts of the tumors. This is the type of changes, right, from CT, depending on the nucleotide that is before and after. For example, lung cancer has a special signature, which reflects like nicotine damage. Melanoma also has a special signature. Here we do find some special signature of mutationals that also have a geographical structure. In this metastasis in the liver, it's different, okay? So that might suggest a different process. Okay? Of course, this is work in progress. Now we need to look at the pathways which are affected there. We are evolutionary biologists, so we care a lot about selection. Okay? Selection is the paradigm, but in recent years, there's been some people saying that maybe not all evolution in cancer is selection. At the very beginning, yes, it is. There is no discussion about it. The first mutations that make a cell become malignant, the establishment of the tumor has to be driven by positive selection. That's for sure. So the first initial cell that outgrows the healthy cells and becomes a tumor has to be selective. But after that, the tumor keeps growing. Is that secondary growth neutral or is under selection? So that's an open question, okay? And there's a big discussion about this. Basically, we can derive the expectation from what is called the set frequency spectrum. The set frequency spectrum is the proportion of mutations at a given frequency. So it's this plot. So you can have mutations with a frequency of 0.1, 0.2, 0.3, and then you plot how many do you see at that frequency. This is a plot that we use a lot in population genetics, okay? The set frequency spectrum, how mutations and their frequencies distribute. So basically, in an exponentially growing cancer population, which is divisions 2 by 2, we assume no death in this derivation, but we could. The expectation is that the frequency of a mutation, or the number of mutations with is inversely proportional to its frequency. It's 1/f. Okay? I don't have it here. So we expect, in other words, to see many mutations at low frequency and rare mutations at large frequency. The more frequent is the mutation, okay, the less common we see that type of mutation. So we expect to see a plot like this. A lot of mutations, okay, at high frequency, at low frequency, and then these are high frequency mutations, very few. And if we plot the accumulative frequency, we should see a line. So we can get our data and check whether it follows this. This is a power law, okay, a 1/f. Whether in our real data we see that a mutation, its frequency, the number of times we see a mutation with X frequency follows this line. And we did this for all the regions, and in most cases we cannot reject neutrality. So the pattern of mutations in our tumor follows the expectations by neutral evolution, except in one case, okay? But, of course, there are statistical arguments here. You might see that this test is not very powerful, which is true, okay? And there are other evidences, on the other hand, that suggest that this history is not that simple. It's not just neutral evolution, okay? Well, we also check population structure. We can really see that this is a principal component analysis, this is a clustering. You can see that there are two well-defined clusters, the primary tumor cluster and then the metastasis, okay? So we see a structure at that level, but not within the primary tumor. We do see a structure between primary tumor and metastasis, but not within the primary tumor. Okay? Basically, in population genetics, to characterize the distribution of genetic variation between populations, we compute something called FST. FST is something about which variance of the allele frequencies is explained by being in different regions, being in different populations in the same region, and so on. And the important number here is that most of the variation here is subpopulations within the total tumor, which means that everything is really well mixed again, okay? But this is a quantitative way of proving this. So tumors are well mixed. We might expect an isolation by distance pattern, which is something very common in population genetics. What is an isolation by distance pattern? It's a situation where if we believe that cells start spreading continuously, we expect that cells that live nearby are closely related genetically. So we expect that siblings, cells, daughter cells, are nearby, therefore, the closer you are in space, the more related you are. Okay? That's a reasonable expectation. That's the null model of phylogeography, isolation by distance. The far away are the populations, the more divergent their genomes are. But we don't see, it's okay. So we don't see a significant correlation between distance between samples, genetic distance, and geographical distance. Okay. Well, to understand in more detail what is happening within a tumor, what we do now is to estimate the clones. So we have bulk sequencing. In bulk sequencing, we get millions of reads, then we need to do all the analysis, and at the end we get allele frequencies. Well, we can try to, tumors are asexual in principle, okay? This is only mitosis. So we can do use some arguments and some restrictions on the variation of allele frequencies to estimate how many clones are there, so how many genetic clones, okay? So the idea is that mutations at a similar frequency, they should live in the same clone. That's the expectation, okay? I'm not going to explain this in more detail. If you want, you can ask me later, okay? But we have methods, many of them, in fact, to infer which clones are present for a given NGS dataset, a bulk dataset. So here we infer 4 clones, okay? We have the healthy, we infer a clone for the healthy cells, a second clone which is an adenoma, a benign tumor that is independent. So we can see that we have the primary tumor in this individual also have an adenoma which is a different origin, okay? And there's a clone. And now we can map the frequency of the different clones here. The white is the estimation of purity. What is purity? Ironically, we call contamination when we have healthy cells in the tumor. Okay? So it's very difficult to get a sample and not get healthy cells from the stroma of the tumor, fibroblasts, whatever. So we can try to estimate that proportion. Okay? And it's very high, but still, we can see a pattern. We can see a pattern where the metastases, they all have the yellow clone, to be simple, okay, which also lives in the primary tumor, but in the primary tumor you also have like blue clone, okay. So this is suggesting something, right? It might suggest that this metastasis also will come from this area of the tumor. That's the most parsimonious explanation, okay, by eye, okay, that this part of the tumor is the oldest one, that spread here and there, okay, and that this might be that. Might be older. This is not totally clear, so what we try to do now is to use statistical methods, not just by eye. So there is something called biogeography, and then we have statistical biogeography, and we have Bayesian methods where we can try to infer the ancestral location, the ancestral geographical location in the tumor. So we have clones, we have a phylogeny. So what I did here is to estimate this is an ancestral clone that we have not sampled. If you know how to read phylogenies, the nodes in the phylogenies are ancestors. So we can predict, this is statistics, okay? We can estimate the posterior probability of the location of that clone. So here, using this methodology, the ancestral location of the first clone of the tumor, the origin of the tumor, is in this region, according to this method. Okay? So we can predict the direction of tumor growth. It happened from here, from the north in this position in the picture, to the south, and from here to the different metastases. Okay? That's what we do in phylogeography, trying to infer colonization patterns. But we can do better. We can also try to estimate time, divergence times, when at least the different clones separate in time. And this is an estimate using again, Bayesian method. We use like a calibration for the mutation rate, for the Okay? This is from the APC gene. There are cell line experiments that suggest this might be a reasonable estimate. And then this is the center of our prior for our Bayesian estimate. It's not fixed at this level. Okay, this is our prior belief. So it's not that it's not fixed. And use some techniques, we can estimate the number of days since these two clones separate. The number of days here is not believable. It's too many days, right? So there's something wrong here. We can estimate the effective population size, which is, this is quite reasonable for a tumor, okay, this number, and we can estimate growth rate of the tumor, just looking at NGS data. And we have here like a positive growth rate, which is not very strong, okay? This is exponential growth rate. But these numbers, this scale doesn't make sense. So we also play with other models. In this case, sorry, I don't have the number, with a street clocks. And we can see that depending on our assumptions of the parameters of the model, we get very different answers about this number of days. So time is very difficult to estimate unless we have a calibration. In evolutionary biology, calibrations are fossils. But here we don't have fossils, okay? So we're still working on solving that problem because I don't believe these numbers. These numbers should be number of days, but are not right. And this is also because we already know now, and we have done simulations, computer simulations, that estimating clones is a very difficult problem. Okay? It's very difficult to infer which clones are there only from the allele frequencies, and that's a problem. Another study, just here this is whole genomes. Okay? This is done by Laura in different locations in 2 patients. In this case, only 3. So basically, we just check the number of mutations. You can see that we get only, less than half in this case, and half of the mutations are shared among the regions. We are talking about hundreds of thousands of mutations that we are able to detect in these tumors. Okay, this is a special type of colorectal cancer, which is microsatellite instability, which they have a lot of mutations. And that's good for us to apply our methods. Well, most of the, maybe you don't see the labels, most of the, indeed, of the variants are in the intergenic regions. Okay, and of course, spacers, and only a few are in introns, okay? So that's expected. There's nothing special here, okay? So the distribution is the expected one. And again, the diversity for all these 2 patients, which we have already 3, is constant, okay? That suggests that tumors are really well mixed. Is this cell migration? We don't know. Is this cell growth? We don't know. This to me suggests that the diversification of a tumor occurs early in time. That's my current interpretation, okay, of this. Of course, we need more patients. Again, these are different, the 3 different regions, and these are different mutations in different driver genes. Again, you can see that some driver mutations, the important ones, the ones that people care, the ones that consortia care, okay, half of them are common, but others are not. Okay, others are not, and these are important mutations. So when people is trying to estimate the number of the frequency of colorectal cancer which have APC mutations, they use one region, okay? So there's going to be a bias in those numbers. But what's the solution for this? Just do a multi-regional study for 2,000 people. Well, that can be done, but it's going to be a lot of effort. But we need to study tumors, not just with a single sample, with several, okay? How about selection? Okay, so in this case, we did measure something which is called the Dnd S ratio. Okay, the ratio of non-synonymous substitutions per non-synonymous site to synonymous substitution per synonymous sites, which should be above 1 when there is positive selection, should be 1 if there is neutral evolution, and should be below 1 if there is negative selection. Okay. There are some caveats about this, but I'm not going to talk about the interpretation of Dnd S in a population like cancer, but let's trust for now that that's the common interpretation. Above 1, okay, we can see that this is missense mutations, nonsense, splicing mutations. In this case, in this patient, they are all well above 1. So there is a clear sign of positive selection, even for splicing sites, which is a little bit surprising to me, okay? Because I will explain a lot of negative selection at the splicing sites. But it's very high. So there is a school of thought in cancer that said this is the common thing. You get a lot of positive selection and no negative selection at all. There's discussion about this. There's a paper last week in Cell proposing that in cancer there is no negative selection, that cells can survive with all kinds of mistakes, tumor cells. Basically, that's the concept. But in the other patient, we don't see exactly the same. Here we see a value which is somehow below 1, well, the confidence intervals are big, in fact. But if this is true, this confirms to be true, so then there is some negative selection. Okay? If there is negative selection in cancer, there is hope, because that means that we can hurt cells, but provoking mutations at certain sites. If there is no negative selection in cancer, that's a big trouble, because then we cannot stop cancer growth. Understand me, just introducing mutations in the genome, okay? Somehow. Well, here you don't see the numbers, but basically, again, we infer the clones, few clones here, surprisingly, just 2. This is whole genome, okay? Whole genome data. We are only able to infer 2 clones. I believe that that's a problem of the methods. But they live everywhere, okay? In this plot, we have the prevalence in different regions. All these clones live everywhere, against total mixing. There is no structure. There is no clones that live here and clones that live there, which is surprising. How about a leukemia? In the leukemia, instead of focusing on the spatial setup, we do have some space in leukemia because we can have samples from the bone marrow and samples from blood. But the advantage of studying leukemia is the capability of getting longitudinal samples in time. So we can get blood different points in time from a patient and follow up the genetic changes. So we are doing that. Tamara is a PhD student taking care of this. So these are the methods. But from this patient, we were able to get samples before and after treatment, which is very nice. In CLL, it's a very slow disease. In many cases, it's asymptomatic. Why? Because the mutation rate is very low in this type of leukemia. And we only detected 14 SNPs. We also sequenced some CD34 lymphocytes that we thought that they would be healthy control, but they are already mutated, which means that the cell of origin goes up, okay, in the hematopoietic lineage than what it was thought before, but that's another story. But basically, we can see that there is a pattern that we can, with this pattern, try to infer the population structure, which makes sense. We can see that, you know, the bone marrow sample is close to the oldest cell compartment, and you have pre-treatment and post-treatment. Somehow, this is a little bit weird that the post-treatment is closer to the bone marrow and then the healthy out there. But this plot here, what we plot is, we have a cell count. We can count the B cells, right? So we know that whether the clonal expansion of these B cells, malignant B cells, is strong or not. So basically, this is a plot through time of the clones we detect. We only detect 2 clones. So this patient went from having a lot of cells, another point, so we talked with hematologists and told us the count dropped down after treatment, so that was a very good sign. But with the after treatment sample, we were able to detect the same clones that were detected before. So we told her, the hematologist, be careful, because this patient, yeah, B cells count went down, which is a good thing from the patient, but the 2 clones are still there. And in fact, 2 months later, this happened, okay? This suggests that there might be some predictive power in evolutionary studies of cancers, okay? This is not easy, it's not gonna be easy, might be hand-waving, maybe, but we might try to predict things looking at clones, okay? Which now with liquid biopsies is gonna be even possible for solid tumors, okay? Looking at ctDNA or CTCs, circulating tumor cells. And that's our next plan also. Well, basically, thank you for bearing with me. A few messages to take home is like, well, there is a lot of intratumoral heterogeneity in cancers. We knew that, but the more we look, the more we see how complex are, just from the genomic point of view. I'm not talking about the functional, okay? If you look at expression level, single RNA-seq, you also see a lot of variation indeed, so it's meaningful variation, okay? We can see that driver mutations are not constant, don't occupy the whole space, so depending on your biopsy, you can miss a driver mutation or a resistant mutation, okay? Which is a driver mutation, of course. There is a lot of mixing, which might suggest there is a hypothesis, at least for colorectal cancer, which, you know, is a glandular tumor. There are crypts, glands, so there are spatial barriers. You know? There is a particular structure with the stem cells at the bottom and the others, which might prevent full expansion. There is a theory called the Big Bang, in which all clones are old. So at the very beginning, many clones appear, and then they have time to mix. Okay? So the fact that we have a lot of mixing and similar heterozygosity levels across the tumors might suggest, in fact, that you get a lot of clonal expansion, many different diversification at the very beginning, at the early life of a tumor. Okay. Still, we detect a low number of clones, but simulations we've done that I have not shown here suggest that this might be artifactual. This might be a bioinformatic problem. Okay. So we need to work harder on that. In CLL, we can see that the clone structure doesn't have to, there are probably not many general patterns, and every tumor is going to be different, and that's also something we learned. But clones can survive treatment, and it's important to follow up the genetic structure of a tumor before and after treatment if we have the capability of getting longitudinal samples. I really believe that cancer is evolution. It's a genetic disease and it's evolution. It's a cancer cell that outgrows healthy cells, acquires a new mutation, outgrows other cells, moves to a different continent and metastasis many times, and only sometimes is able to see the metastasis, outcompeting the cells in that organ. It is evolution. It is somatic evolution. So it makes sense that evolutionary techniques, evolutionary biology, which is hundreds of years old, can be able to say things about it and complement others' point of view. And that's the direction our lab is taking, trying to bring the world of population genetics, phylogeography, phylogenetics to cancer. Okay? But in population genetics, in phylogeography, we work with individuals. And here individuals are the cells. So to really exploit, to really have power to estimate quantitative evolutionary parameters like growth rate, wouldn't that be an important parameter to estimate growth rate from a single biopsy? I believe it would. We do need single cell techniques, and we are working on that. And hopefully, this will be easier next year, and we'll have some single cell genomes to present with more accurate estimates. Okay, so that's, to finish, I just need to acknowledge people in my lab here, okay, that work in this and other things that I didn't show you, and getting samples when you go to Cape Verde and do some snorkeling is very fun and relatively easy, but getting samples in the cancer setting requires that, some kind of friendship with a pathologist. And it's very difficult. So there is a lot of limitations about the sampling that we can make. So I really want to thank these two people, Jose Manuel and Carmen, because they are great and they've been really helpful providing the samples. And other collaborations for different aspects. Now we are trying to measure the environment using immunohistochemistry, because the whole story cannot be explained without the environment indeed, and trying to understand methylation and other levels of the whole problem. And that's everything. Thank you very much.
Thank you, David. Any questions?
A few questions, but short. First, in one of the previous talks, we learned that there are many macrophages invading solid tumors. When you had a single cell techniques going on, how are you going to distinguish between normal macrophages and transformed tumorous cells?
That's a posteriori is very easy, just looking at the genomes, because the macrophages their genome will look like a healthy genome.
The standard one, all right.
So, no, that's not a problem.
It would not be.
And we do fact sorting. Okay, so when we do single cell, we do sorting. Okay, with LGR5, blah blah blah. So which is imperfect. But so we distinguish a priori, but we can always distinguish for sure a posteriori, looking at the genome, but then this money that you throw away.
Yeah, all right. And now on the issue of the maintenance of the clone structure of a tumor a long time and through therapy, I don't really understand if there is a high rate of mutations going on just in the mitosis that these cells undergo. I would expect an evolution of the spectrum of the...
But it's a question of power. We don't detect that. Okay, so these mutations are so rare because you have, in a tumor, you have 10 to the 14 cells, something like that. That's a big number. And when we sample, we might be sampled 10 to the 9, 10 to the 10. And then we do sequencing at 100, I don't know if you're familiar with NGS, so we sequence at 100x coverage. So we can only detect alleles which are at 5% or above normally. This is a generalization. So most mutations are rare and we don't see them. This is what So these are main clones, like main clones, and inside maybe there are many mutants. So it's coherent, what I'm saying. It's compatible. It's just a question of power. It's your, your microscope. So if you look at a tree, the cells are the tip, and we just look at a slice which is at the earlier branches, older branches. But it's a good question.
But will you detect that microevolution when you had single cell?
Exactly.
All right.
That's the point. With single cells, we can detect a mutation as rare as being in one cell. Well, that's not true. It's impossible to detect a mutation in a single cell from an error. So what we do with single cell data is just to trust mutations that we see, that we call, because this is statistics, okay? Everything this is statistics, really, that we see in at least 2 cells. Apart from other filters, we require mutations appear in 2 cells. So therefore, we can detect very low frequent alleles with single cells. And there are other techniques for detecting rare alleles in cancer, but it's possible. But we will never, ever detect all alleles in a tumor, because that's impossible. We will need to sequence, billions, European billions of cells.
Thanks for your nice talk. My question is about the mutation rate, because you're assuming that it's stable, but actually most
No, I didn't assume. So, sorry, can I interrupt you? So I put something there called a relax clock. The relax clock is a model.
Yeah. But
so we assume that that rate I put there, this is like the mean of a prior distribution from the variation of the mutation rates. So I don't assume it's fixed.
Okay, but that's actually, because that was only the beginning of my question. My question is about susceptibility genes. So actually most of the genes that cause cancer actually is risk, because what they do is that they affect, many mutations affect repair genes. So actually what you've got is that you've got a pre-state of high mutational risk, because you've got a mutation on nucleotide excision repair genes or mismatch repair genes. So already you are prepared to mutate very quickly. So in a way, what you've got is that depending on the germinal mutation, or even if it's a somatic mutation in these genes, the way you're going to evolve, your genome is going to evolve, is already in one direction. Because if it's a mismatch, it's going to be a slippage or whatsoever. If it's in nucleotide excision repair, you're going to be prone to certain types of mutations. So that's what I'm asking. So in a way, you are primed for going to a direction. Well,
there are different angles to answer your question. One is that what you are mentioning is called the hypermutation phenotype and things like that, the ApoVic route, right? So there is a debate in the cancer community about whether it's true that we see a lot of mutations or we see that a cell divides very fast, because it's not possible to distinguish mutation rate from time. You can have many mutations either because you get a high mutation rate or because that cell divides like crazy. So mutation rate per generation, per mitosis. The fact that the word direction is very gross, right? So what is a direction? It's true that there are some mechanisms like you're talking about. So we have that in MSHL cancers, in these colorectal cancers, right? Patients, methylation of BRAF, things like that, so you get higher mutation load. But that, then, mutation is just the substrate for selection. So it doesn't preclude different, it might take you to some direction regarding your functions, I guess that's what you mean, right? Your functional capabilities. But
depending on the gene of the DNA repair that is affected, the type of mutations that are going to appear are one or the other, because the DNA repair mechanisms are altered. That's the point.
No, that's if you remember, I put something called mutational signatures, right? Which is a way of characterizing the type of mutations that we get. And this we have now like 20 mutational signatures, with, signature 1, signature 2, signature 3. Some are associated, as I say, to lung cancer, to melanoma. And we know that this can change through time. And in fact, the mutational signature of the liver metastasis here was different. But I agree with you, but I don't see any problem for inferring evolutionary parameters. You just infer what you have. So some people, I get this question sometimes, and I guess that's a little bit of confusion, saying, well, but your patients might be treated. And I say, and what? I am going to study the evolution of that patient. whether it's treated, whether it's untreated, whether it has a mutation in, P53 or it has a mutation in a repair gene, it doesn't really change my way of studying. What you're saying, and it's true, is that this is very idiosyncratic. Every tumor is different. Yes, I totally agree. Depends on many things. Depends on things like the one you're saying, right? Which mutational pathway is activated, but depends on other, how many oxygen do you have, how much do you smoke, gender, germline mutations, many contexts. So we do need to move into personalized, precision medicine, personalized, and to understand the personalized evolution of single patients, and that's what we're trying to do, in fact. I don't know if that answered your comment. I don't know what your question was, actually.
David, here, on this side also. Fascinating talk. So basically what you get with your analysis is like a snapshot, and then you try to infer how this has evolved. Now, could you imagine a model where you actually could experimentally let evolve this system and then take time points, a little bit like you did with your leukemia, but in a, even more experimentally suited, because I think the dynamics of the mutations will be crucial.
Yeah, no, that can be done, and it's done in cell lines, in xenografts, mice, so you can get all the samples that you want in principle, with patients only in liquid tumors, unless, as I mentioned, you have liquid biopsies. Yeah, this is we just have 2 points in time, but you could do 10. That depends on the pathology, but it's a compromise. The logistics matter a lot here, logistics, because you work with patient data, and then the clinicians are not worried about me, are worrying about the patient. So I cannot say, sample this guy again. Well, maybe blood, I can suggest whether it might be useful. But yeah, we will learn a lot. Most evolutionary biology is like you mentioned, we get a snapshot, we get the human chimp orangutan genome in this century, and we try to infer the past, what happened in Africa 2 million years ago, 1 million. So we are used to that, to work with the present. But in cancer and in other organisms, we do have the opportunity to sample several points in time. But, that depends on the patient, the doctor, the type of cancer, but indeed, that's much more informative. So liquid biopsy will help a lot in that regard. We will be able to see evolution on time with circulating tumor DNA, or with, well, we look forward to that.
Hi, David, beautiful. I have 2 questions. One is very short answer. Do you disregard mitochondrial DNA in the analysis?
No.
So, and the clonal expansion, so the clonal definition only by mitochondrial DNA is identical to the others. And the other, and just I put on the table, the other question is that I understood that you are considering the clones and the evolution of the tumor as the individuality of the clones being less or more successful. But can you model the possibility that there are symbiotic interaction between clones in a way that this really enhance the aggressivity of the cancer?
Yes. Well, first question, we don't have mutations in the mitochondria. Mitochondria is 13 kb, no? Or something like that. It's very small. So there are mitochondrial mutations in some cancers, but we don't see them. In these cases, it might be in some patients you might have, but you are going to have 1, 2. So we just want the, thousands. We don't see them in these cases, as far as I know. So these mitochondria are identical in principle, okay, with that. But we can go and double check on that. But okay. So the second was, yeah, there are hypotheses about clones cooperating, about clonal cooperation, cell cooperation, which is maintaining phenotypes that like a global population phenotype, because you can have compensatory mutations in different clones. So yes, absolutely. We are not looking at that. We cannot look at every angle. So we are looking at the ones where we are more expert, which is the evolutionary history. But these ideas are out there about clones cooperating.
Very nice, thanks a lot. I have a question. Did you look at the heterogeneity at transcriptional level and epigenetic level to see how much this match? Because
we are, that's on the, it will happen. We are already, in fact, this morning I got methylation data, but just for a few sites that we want to use as clocks, mitotic clocks. These are loci that do not express in the colon, and therefore they accumulate mutations, we know, because of different previous literature, according to time. So we're looking at that. That's very interesting. RNA-seq, single cell is much easier with RNA than with DNA, much. 99% of the single cell research is about RNA. You have machines, you have free dime, you have the C1, all kinds of things. But we have not started there. Why? Because we just want to focus on something we really have experience, which are statistical models of DNA substitutions. But we will move a little step ahead with the methylation, with measuring epi alleles. Okay, like CPGs, sites are on, off, as 0, 1s, and we will try to correlate. Indeed, the whole picture should make sense. For RNA-seq, it has a lot of problems also. Okay, about allelic imbalance, about missing alleles. I just listened to a talk last week about really powerless estimates of allele frequencies with RNA-seq. So at some point, we will be happy to look at that, but it's not in the immediate agenda. But it is important to look at the whole thing at all levels at the same time. But we are limited, and we do it things step by step.
Congratulations on your interesting talk. That's why you're having so many questions here.
Thanks.
So, I'm particularly interested in cancer stem cells, and it seems to me you haven't told us much about it. It would be very interesting to be able to detect them and even quantify them.
We are doing
that. Tumor.
We are doing that already. So the postdoc in my lab, I never had a wet lab until 3 years ago, so I'm a computational guy. So I had to hire a cell biologist, of course, and she is a stem cell biologist. So she's sorting stem cells. You might be, of course, the stem cell hypothesis, which means that, the cells that feed the tumor are the stem cells, the ones that reproduce. So we plan to test the hypothesis at the single cell level with the genomes. But as I'm saying, single cell whole genome is difficult technically, and we are working on that.
Yeah, it's much more than a hypothesis, since in most, if not in all, tumors, they're being detected little by little. And what I think is that at the genomic level, there won't be much differences regarding comparing them with other cells in the tumor.
But we can check the history. So our point is not about I understand what you mean about function, but we can check whether they form separated clusters along the history of the phylogeny of the tumor. No. So we can see whether they are intercalated. So we have stem cells, giving cells and cells, or you have a separate clusters that initiated one. So we will be able to test the stem cell hypothesis from a phylogenetic perspective.
Single cell sequencing will be helpful on this topic, I believe. Thank you.
Thank you.
Thank you very much. Miguel has a message for you before you leave.
Yes, so just before we leave, at 3:30 we'll begin the specific symposia. And just to remind you, the genetics one will be here in the auditorium, cell biology in a room that's just in front of registration, and developmental biology in a room that's downstairs. Okay, so that will start at 3:30. Now there's also going to be the poster sessions. Remember that there's going to be prizes for each society specific for posters and talks, so try to identify clearly which society you are a member of when your posters. Okay, so thanks a lot, and thanks to the speakers for such a good session today.